Hey everyone, today I'll be talking about the application of the technology CRNAseq to get insights into microbial communities and HPV infections. About the cervicovaginal microbiome, in healthy cervical conditions, the microbiome is characterized by Lactobacillus dominans, and a change in this composition causes dysbiosis. An example of this is bacterial vaginosis, which is characterized by overgrowth of Gardnerella vaginalis and depletion of Lactobacillus species. The microbiome is also characterized of community state types, or CSTs, which were first described by Rabel et al. In 2011. And they are classified based on Lactobacillus dominans. For instance, if the microbiome is dominated by Lactobacillus crispatus, is typical of CST1, Lactobacillus gaceris, CST2, Lactobacillus iners, CST3, Lactobacillus jensenii, CST5, but if it is characterized by a diverse microbial population, that is CST4. The classification of CSTs is usually performed by unsupervised clustering analysis, but there have been an increase of studies classifying CST for up to 9 groups. So there's a need to a consensus for classification of CSTs. For this purpose, there was a recent development of the tool, Valencia tool. They can classify CSTs using a huge data set of microbiomes. But the downside of this tool is that it requires 16S ribosome RNA gene sequencing data, which provides a species level profiling for a limited amount of species. And also we know that for 16S, there's also problems with PCR bias. And for instance, here, with the use of, for the use of different variable regions from the 16S gene, if we use variable regions 1 and 2 for microbiome profiling, we get a different CST assignment than we use variable regions 3 and 4. So there's also other technologies for microbiome profiling, like shotgun metagenomics, but this technique is quite expensive and requires specialized skills for that analysis. So to overcome this, we also developed this technique called circular probe-based RNA sequencing, or CRNAseq, which employs SMIPs, or singular molecular inversion probes, that is able to target a specific microbial species and profile the microbiome. So with the technique, we target multiple viral regions of the 16S ribosome RNA gene, and with this, we are able to get to the species-level annotation of microbes. Our panel consists of around 321 microbial species that are relevant in the microbiome environment. And we can perform profiling of the microbiome at both DNA and RNA level. It's important that we also, with a technique, we can perform high-throughput sequencing, and it's very accessible for data analysis. And for our field of looking at the relationships of microbiome and HPV, the techniques can perform in a single run both HPV detection, genotyping, and microbiome profiling. In terms of the association of the microbiome and HPV, there has been studies describing that the CSTs associate with protection against pathogens and viral clearance, and also with HPV infection and persistence. Even the virus infection causes dysbiosis and promotes different, like CST IV during disease. There's also association of CSTs with low and high-grade cervical lesions, and there's an increasing field of looking at microbiome targeted therapeutics to target the microbiome and promote these Lactobacillus dominans, and in terms of HPV infection, promote viral clearance and disease regression. So in this study, we validated ciRNAseq. We also look at the association of CSTs with HPV infection, and there we found the potential of the technology to classify CSTs. And we therefore wonder whether by applying high-resolution microbiome profiling, we will be able to classify the classical CST and perhaps discover new CSTs. For this, we use a large cohort of cervical smell, around 541, from Dutch women participating of the population-based cervical cancer screening program. And here we apply the technology to profile the microbiome and perform unsupervised cluster analysis to classify CSTs. As you can see in unsupervised cluster analysis, we observe the classical CSTs in our profiling. And as the first that you can see is two main clusters that are characterized by microbial diversity. You can see that there is more diversity in the left cluster than the one in the right. That is more characterized by Lactobacillus dominans. And there we can see that some species level associations with these dominants in CSTs. And also we found that in these major groups of CSTs, there were certain species that cause a separation of communities. And for this, we classify CSTs in A and B subgroups, depending on the associations of a species like Lactobacillus acidophilus, Megasphaera genomosp type 1, Lactobacillus iners, and Lactobacillus jensenii. We also, to visualize these differences in CST classification, we look at the microbiome composition by the classical CSTs. And as you can see, these CSTs dominated the PCA axis, and specifically some species. When we look at the classifications that we found, you can see that these communities had specific subgroups in the PCA, and therefore were indeed communities. We look also at the number of species and Shannon's diversity in these CSTs, and we found that in the B subgroups of CSTs 1, 3, and 4, there was a higher number of species than the A subgroups. By looking at the bacterial relative abundances and CSTs, in CST1, we observed there was a decrease in abundance for Lactobacillus crispatus in CST V, and there was an increase in abundance for Lactobacillus acidophilus and Lactobacillus inner. In terms of CST 3, we also found there that there was an increased abundance of Lactobacillus acidophilus in CST IV-B when compared with the CST III-A. Additionally, we found that for the species Lactobacillus jensenii, there was an increase in abundance for in the CST I-B and III-B when compared with the A subgroups. And Lactobacillus acidophilus was also found highly abundant in CST V. For CST IV-A and B, we found that there was an increase in abundance of Megasphaera genomosp type 1 in the B subgroup when compared to the A subgroup, and there was a significant lower abundance for Gardnerella vaginalis in CST IV-B when compared to IV-A. To validate these CSTs, we also look at the cohort with HPV negative women and HPV positive women with either without cervical abnormalities or with either low or high grade cervical lesions. We look at the classical classification of CSTs and the classification with the novel subgroups. And we found associations with the different groups. So CST V was associated with HPV negative conditions, and we found a small association of CST I-A with HPV positive condition, but without cervical abnormalities as CST IV-B. And we found an association of CST IV-A with low-grade cervical lesions. For this study, we also proposed a model for microbiome dynamics. We believe that perhaps these CST subgroups represent interstate of communities in the microbiome, and also during disease. So during the progression of HPV infection, there is an increase in diversity in terms of CST4 and a depletion of Lactobacillus species. So in summary, we characterize CSTs based on high-resolution microbiome profiling and determine their clinical relevance through our HPV infections. Our classification suggests microbial dynamics occurring in health and disease by proper longitudinal studies are warranted, and we are working on that right now. Our data emphasize the identification of commonly overlooked species like Lactobacillus acidophilus and Megasphaera's genomosp type 1 that we believe are relevant for cervical health, microbial relationships, and dynamics, and which require high-resolution microbiome profiling for adequate annotation. And although we analyze CSTs in the context of HPV infection, more studies are required to explain the importance in cervical health and disease. So finally, I'd like to thank all my supervisors at Radboud University Medical Center and the REMOS, where I'm doing my studies, the Robbie and Rose Foundation that funded the study, the Microbiome Virtual International Forum for the opportunity. And if you'd like to read more the complete story about CSTs and HPV, the study is already published in the NPJ Biofilms and Microbiomes journal. And here's also a code with the link. Thank you very much.